What's up, Wolverines? Today we're going to go ahead and pick up some other odds and ends and related to genetics that we haven't been able to get to yet. So we're going to go ahead and go over genomic imprinting, which is a form of epigenetics. We'll talk about what epigenetics it is. We'll go over genetic counseling, the value of genetic counseling and what we can use it for, different types of genetic testing that we can go ahead and do that can help inform genetic counseling. And then we'll take a look at sex determination and karyotypes. When we talk about sex determination, we're starting to look at what makes it male, what makes it female, because the process is not exactly the same for mammals as it is for other organisms. So we'll take a look at mammals like us, but we'll also go ahead and take a look at some of our insects, butterflies, and chickens, these types of things. So you can see kind of some of what's out there. And then lastly, we'll look at those karyotypes, as I mentioned. So we can use that as a tool to go ahead and look for chromosome number mutations, non-disjunctions that we talked about before. That produce aneuploidies. We can also go ahead and take a look for chromosome mutations if the size is different, it's either too large or too small. These types of things. This is actually one of the ways that they can actually be sure about the offspring's sex before the child's actually born is by looking at the actual chromosome. So we'll go ahead and take a look at some of those things. Let's take a look at our essential questions. Number one, what is imprinting and how does it work? Number two, how is gene mapping done? Okay, how do we know where chromosome genes are on a chromosome? Number three, what is the purpose of genetic counseling? Number four, how is sex determination in other species different than XY sex determination in mammals? Remember, we've talked a little bit about XY, but we'll kind of clarify that before jumping into the other types of sex determination. And lastly, what's a karyotype and what can a karyotype show us? So let's go ahead and start with genomic imprinting. Now, if you think about the word imprinting, it means kind of to leave a mark in. And so when we talk about genomic imprinting, we're talking about leaving a mark on your genes. The phenotypic effects of some mammalian genes actually depend on whether they're inherited from the mom or from the dad. This is the phenomenon that we know as genomic imprinting, and it's a part of a much larger concept known as epigenetics. Epigenetics is a process where we look at how what you inherit impacts your phenotype other than just the actual nucleotide sequence. Okay, so what we've discovered is that the expression of your DNA is not the only thing that impacts how you grow and develop, but things in your environment and what that does to your genome also impacts it. So, just as cells inherit genes, they also inherit instructions that tell them which genes to turn on, which genes to turn off, and in what tissues and how much. And so, these types of what we call epigenetic phenomenon are important because they regulate when and at what levels certain genes are expressed. Let's take a look at this a little bit and give you a couple examples so we can better understand how it works. So, when you get the egg from your mom, that egg, the DNA that's inside of it, is actually imprinted upon. Your body somehow knows that that's from your mom. Likewise, the DNA that came from your dad, the paternal imprint, is placed on the chromosome in the sperm. So your body knows what DNA it got from dad. Most of the times when we've looked at inheritance previously, we just say, I've got this complement of genes found on these particular homologs. But what we're finding is that imprinted genes, maternal imprinting and paternal imprinting, actually does impact development. Males tend to show more of the paternal genes, and females tend to show more of the maternal genes. This may have something to do with the biological advantage of if your mother was able to go ahead and produce offspring, you're probably more likely to do the same if you have some similar traits. Okay. So when we start taking a look at how somatic cells are expressed, when we start going ahead and taking a look at phenotypes, there may be some phenotypes that are more advantageous to one particular gender. And then when we go ahead and pass those on, These different imprinting processes um, go ahead and pass on to the next generation, but then they seem to get wiped when they go to the generation after that. And so you have new imprinting every generation so that it just seems to be following the maternal and paternal imprinting. So why does this matter? Can you give an example? So let's take a look at one particular mutation that is related to gene imprinting. So what we're going ahead and looking at here is a particular chromosome, and we have a deletion mutation, and it's the exact same deletion mutation on both the paternal and the maternal. Let's take a look at the impact. Now, in some cases, not in all cases, but in some cases, the gene mutation um, works out differently if it's from mom or if it's from dad. Okay. There are two disorders, both very different disorders, but both caused by the very same deletion on chromosome number 15. Now, if you get the mutation from your father, so on the paternal on the left there, this gives you what's known as Prader-Willi syndrome. If you have Prader-Willi syndrome, you exhibit mental retardation, obesity, short stature, unusually small hands and feet. But if you get the exact same mutation, not from your dad, but from your mom, you end up with a disease called Angelman syndrome. And Angelman syndrome is quite different from Prader-Willi syndrome. Gives you uncontrollable laughter, jerky movements, motor and mental abnormalities. What comes to my mind when I think of Angelman syndrome is the character of the Joker from the Batman comic world. Okay, so two very different diseases, both caused by the exact same mutation. The only difference is if you got it from your mom or if you got it from your dad. So pretty unique when we start looking at some of the impacts of epigenetics in terms of the expression of these particular genes. That also leads to what's known as gene silencing. When we start looking at imprinting, it's not always parent-specific imprinting. We can also go ahead and take a look at what we call silencers or activators. Certain genes can be turned on or certain genes can be turned off. One of the most common ways to turn a gene off is through what we know as DNA methylation. And it's pretty well understood at this point, after many years of study, that essentially what happens is DNA methylation turns off a gene's expression. So it puts the chromosomes that are found in the chromatin into essentially a like hibernation state. And what they do is they add a little methyl group to the cytosines in the DNA. So remember cytosine, guanine, adenine, thymine. So the cytosines get tagged with this methylation, and it stops them from going ahead and being produced as proteins. Now, this changes the state of the expression of any genes. So if there's methylation in the region, it gets turned off. And in this way, either the maternal or paternal gene may be silenced, and it doesn't really matter if it's a dominant gene or a recessive gene, it can still get turned off. Now, if it's a recessive gene, you wouldn't notice it, but if it's a dominant gene, you may end up showing the recessive trait from the other parent, even though you have the dominant allele. So this is where this kind of epigenetics impacts it, is that we're seeing a combination that normally wouldn't happen because that's not what dominance and recessiveness says happens. But because of the environmental impact causing a methylation, that gene gets turned off, and so you express something else. Okay, so we'll talk about this. There are examples like bar bodies, for example. If you've ever seen a calico cat, stop and think for a minute. Are calico cats male or female? Now, in the vast majority of cases, calico cats are female. Very rarely do you get a male calico, and that's because these different expressions are due to gene inactivation on the X chromosome. Now, for males, you can't inactivate an X, otherwise the male is missing a ton of important genes. However, if you have a Klinefelter's cat, they now have two X's, and they can go ahead and inactivate one of them and get the calico tortoiseshell coloring. Okay, so the X inactivation that we just talked about with the bar bodies is a good example of gene silencing. All right, so you're kind of getting an idea of there's more to inheritance than simply I got this gene from mom, I got this gene from dad, and this one's dominant or this one's recessive. We have codominance, we have incomplete dominance, we have lethal alleles, we have polygenic traits, we have multiple alleles, and now we're throwing in epigenetics, where the environment actually can dictate some of the outcomes of what genes are turned on and off. We also have epigenetics and the fact that parent of origin genes can be activated or inactivated depending on what gender the offspring is in, as that can help befit them be more successful in finding a mate. The other thing we start looking at is how do we know what genes are on what chromosome? If you remember last class, we started talking about linkage, right? Chromosomes that are found on the same chromosome. So how do we know that? This is done through a process known as chromosome mapping. Now, in most cases, the further apart any two genes are on the same chromosome, the greater likelihood they have of having a crossover mutation. Do you guys remember crossover mutations? Hopefully you do. This is when you have your tetrad in synapse during meiosis. Now what happens is when you end up with those four chromatids so close together, the inner two chromatids sometimes get overlapping. Okay, and that overlapping, if you remember the structure of the chromosome, is like getting a slinky tangled. And it's really hard to get apart. So what your body does is it cuts it and puts it back, but sometimes it swaps genetic information. Now, when we start looking at that point of chiasma, it matters where that is in terms of chromosome mapping. So what we do is we go ahead and do tons and tons and tons of this copying to go ahead and look at the stages of meiosis, and we look for how often these two particular genes get separated during a crossover. So if we go ahead and take a look at a chromosome, here you can see a chromosome with gene A and gene B. Now, if we go ahead and do this 1,000 times, we can find the percentage of time that there's a crossover in between them. Let's say we said 26.8% of the time you end up with a crossover mutation between them. Well, we would convert that then into what we call chromosome maps. So you would be 26.8 map units between point A, gene A, and point B, gene B. We can go ahead and add these all up, and we found that they become very reliable in terms of establishing a pretty good idea of what region of the chromosome a particular gene is found on. So let's say in the map below at the bottom, you see a crossover frequency of 10% between A and C, and we would identify that as 10 map units apart. You see a crossover frequency of 12 between B and C, 12 map units apart, and you see a crossover frequency of 22% between A and B, 22 map units. And now if you add up 10 map units and 12 map units, that equals the 22 map units, and that pans out really cleanly. And they've had really good luck doing this, and so it's given us a pretty good idea of not only what chromosome it's on, but where on a particular chromosome a gene is located. If we understand where genes are located on a chromosome, if we have an idea of dominance and recessiveness, including all the variations, codominance, incomplete dominance, etc., if we understand the role epigenetics can play in determining the outcome or the phenotype of genetic crosses, we can really provide some good detailed information to families who are at risk for particular diseases or require other types of genetic counseling. So what is genetic counseling? Genetic counseling is an analysis of the risk of producing offspring with known gene defects within a family. This is typically coming into play when we start looking at families that have a particular genetic disorder in their family history, We start thinking about things like Huntington's disease. Maybe you start looking at epilepsy. Maybe you start looking at other inheritable genes. But we can also start using genetic counseling when we start looking at those that are having offspring when they're in what they would consider advanced maternal and paternal age, because then you're at higher risk for producing atypical gametes, which might go ahead and change the outcome of the phenotype of the offspring. So, what do the counselors do? They have a whole list of things they can do here. They can identify families that are at risk in the first place and investigate the problem that may be there. They can interpret information about a particular disorder, analyze inheritance patterns using things like the pedigree we talked about last class, and they can help review available options with the family so they can make the best decisions for them. When we start looking at these types of counseling, we can go ahead and start looking for the risk of particular birth defects or genetic disorders, and so we can start trying to mitigate them. We can predict that there's a high likelihood for certain diseases and give treatment to those, or at least test to see if they have the disease early on to get them the treatment they need. About half of the cases of childhood deafness are the result of an autosomal recessive disorder. If we can identify the problem early and get them the appropriate treatment, they don't have to deal with a lot of the problems associated with that early childhood deafness in terms of speech lag and things like this. And then lastly, we can go ahead and start have testing done prenatally. Pregnant women can go ahead and do it, and this is actually a common test that they have for those of advanced age, especially maternal and paternal. Um, and we can look for particular inherited trisomies, like Down syndrome, for example. Here's trisomy 21, and look for other genetic abnormalities such as neural defects. Trisomy 18, that's the one we talked about, looks like gummy bears. So we can go ahead and kind of get an idea of what to be prepared for as parents that the individual offspring may carry. Now, one of the things that we do, not just with a karyotype that you saw on the previous page, but with several different types of tests, is try to identify the presence of defective genes. There are more and more DNA tests for identification of these types of genes, and they're constantly working on more. And so, while screening for many genetic disorders is now recommended, the use of pre-symptomatic testing for adult onset disorders like Alzheimer's is still considered pretty controversial. It's that idea of, do you want to know that you have a high likelihood of a particular disease later in life? Yes, maybe, because that allows you to go ahead and prepare for it, but also no, because then there's that kind of idea where it's like a train's bearing on you and bearing down on you, and you know it's going to hit you, but you don't know when. Um, and so this is still pretty controversial. And as we talked a little bit about last week, we're going to try to get in some of these questions of bioethics later in the week. Okay. While there are benefits to knowing this information early, because for some of these diseases like Alzheimer's, we don't really have a treatment for them, are we dooming these people to just kind of feel like there's nothing they can do about their life because I'm going to get this disease? Okay. Um, so it's a good thing to start thinking about, how would you want to know, or would you want to know at all? Okay. What kind of things or genetic tests may include? Um, there's a wide variety. You can see them here in this table, but everything from biochemical tests for particular gene products, an enzyme that's produced, or some other protein. This is the kind of thing we can start looking in terms of carrier screenings to see if you have it, and then we can think about the likelihood of it being passed on. Um, we can go ahead and do certain types of pre-implantation genetic diagnosis by using microscope examination of fluorescent stained chromosomes. Okay, do we find a particular sequence that is at high risk for things, certain sorts of genetic flaws? We can do this before we go ahead and implant uh, embryos for IVF. We can go ahead and do prenatal diagnosis where they go ahead and take some amniotic fluid and go ahead and look for things like Down syndrome or other trisomies. Or once the child is born, we can go ahead and do some sort of pre-symptomatic testing. Okay, or even for adults, if we're worried about something like, say, Huntington's disease, you can take a blood test to determine the likelihood or whether or not you have some risk factor for Huntington's disease or cancer. All right. So, regardless of what you're doing these biochemical tests looking for enzymes, you're looking at the fluorescent stained chromosomes, or even examining the sequence itself of the DNA, A, T, C, G, so on and so forth. We can use these tests to help us know whether or not a particular gene is present. In the example of sex-linked dominant forms of rickets that we talked about last class, the ratios affected children can be determined in the phenotype and genotype of the parents is known. So in this particular case, we're going to take a look at a family where the husband has a um, rickets, right? He has this X-linked trait for a certain form of rickets. Now, in this particular case, the prospective parents would be advised that there's a 50% chance of having an affected child. Uh, the male, if he has a son, would be fine. But if he has a daughter, because it's a dominant disorder, every daughter he ever has is going to have rickets. Okay, so we can go ahead and look at the gamete formation. You can see if we look at independent assortment and segregation, um, female is a normal woman. It can only give normal X's to males and females, but the husband is going to give Y to males and the affected X to all of his daughters. So all the daughters will have rickets. Okay. So let's take a look at some other disorders that we use this type of genetic counseling to help us predict the likelihood and whether or not we want to continue maybe seeking out our own children. There are a whole bunch of inherited disorders caused by dominant alleles on autosomes. Now remember, autosomes are those that are 1 through 22, not the X and Y like the rickets we saw on the previous slide. Okay. There are many examples, and they're evident in both heterozygous and homozygous dominant individuals because you only need one copy to get the disease. One example is diabetes insipidus. This is where you don't make enough ADH or antidiuretic hormone, and this leads to production of large amounts of urine, extreme thirst, and it can also, however, be produced by an X-linked gene. So there are different versions of it. Another one of these, probably the one we've been talking the most about, is Huntington's disease, which causes involuntary movement of the face and limbs with general mental deterioration over time. The beginning of symptoms typically varies, but for most people, it's between 30 and 40. But I've heard of people that show symptoms up into their mid to late 40s for the first time. We also have autosomal recessive disorders. Um, these you have to have two copies of in order to go ahead and show the disease. And when we start looking at this, this includes things like cystic fibrosis. The young man here is being checked out for cystic fibrosis. They treat them with inhaled drugs and physical therapy to try and help clear mucus from the lungs because cystic fibrosis produces a thick mucus that can often lead to pneumonia and emphysema. Cystic fibrosis, or CF, is probably the most common lethal genetic disorder in childhood and has an incidence of about one case in every 3,700 live births. The next one, a lot of people think sounds funny, but there's nothing funny about the illness. This is what's known as maple syrup urine disease. If they have this gene combination, it causes mental and physical retardation produced by a particular block in amino acid metabolism, so they can't break down a particular amino acid. Because of this, isoleucine in the urine produces the characteristic maple syrup smell when they urinate. All right. And then we get to Tay-Sachs disease. Now, Tay-Sachs disease is a lipid storage problem, which over time eventually leads to paralysis, mental deterioration, blindness, and death, typically by the age of 3 years old, because they're storing fat in atypical locations. So rather than putting your fat in your stomach and around your hips, they will put fat in and around their brain and start closing off things like optic nerves and stuff like that. So it's pretty sad disease. And some of these mutations that we're talking about that cause this are pretty small. Okay. So if we take a look at something like cystic fibrosis is probably one of the simplest diseases to go ahead and cause. The most common allele that causes cystic fibrosis is missing 3 DNA bases, losing 1 amino acid. That's it. So the amino acid phenylalanine is missing from the CFTR protein. And because of this, okay, it's missing a protein ion channel and it cannot communicate properly. And we can't transport the CFTR where it needs to go. And the cells in a person's airways become unable to transport chloride ions. This is part of why they become so clogged and thick with mucus. So it's pretty crazy how something that simple can have such a profound effect on the individual with mutation. Okay, so we've now looked at the value of genetic counseling. We talked about some epigenetics. We want to go ahead and start finishing up with sex determination and karyotypes. So let's take a look at sex determination. Now, we've already gone over this idea that in mammals, you have XY sex determination. If you have a Y, you're male. If you don't have a Y, you're a female. And the male is what we would call, this is a new term, the heterogenetic sex. Hetero means different. So heterogametic means 2 different gametes for sex. So we have the X and the Y, because remember, females will have 2 X's. And this is going to be true for all of our mammals. They have XY sex determination, but it's not always the same. Okay, and the heterogametic sex is not always the male when you look at some of our other species. Okay. All right, so let's take a look and start with XY sex determination. This is based on the presence of the Y chromosome. If you don't have it, you're going to develop into a female. Mammals, including humans, the fruit fly Drosophila, so Drosophila melanogaster, which we do a lot of genetic tests on. Some of our dioecious plants. Dioecious means separate male and female plants. I know you probably didn't think about the fact that there are male and female plants, but there are. If you've ever gone to an orchard for many types of trees, they'll have one male at the end and the rest of the row will be females to go ahead and produce fruit. Okay. Kiwi fruit's a good example of this when we start thinking about this. And then they reproduce not through typical fertilization methods like we think of, but through pollination. Okay. So females in this case are homogametic, meaning they have 2 of the same chromosome. The male then is the heterogametic sex. It has the X and Y. And your primary sex characteristics are inherited mostly on the X gene, except for maleness, which is determined by the Y chromosome. Now, again, that's primarily for mammals, along with your Drosophila, some dioecious plants. We have a very different inheritance pattern when we go ahead and start looking at birds, for example. Birds exhibit what's known as WZ sex determination. But in birds, the heterogametic sex is the female. So she has 2 different sex chromosomes versus the male who has homogametic, 2 of the same sex chromosomes. Okay. So when we go ahead and take a look at this, you can see the female has WZ. And so if they go ahead and pass on the W, it becomes a female. If they pass on a Z, the male can only give a Z, and so they end up being male. So it's important to kind of keep this idea in mind. In WZ sex determination, the female determines the gender of the offspring, but in XY sex determination, the male determines the gender of the offspring. Now, this occurs, as I mentioned, in birds, but also in butterflies, moths, some fish. Okay. The next type that I'd like to talk to you about is even more different than that. This is what's known as XO sex determination. In some insect orders, the female has 2 similar sex chromosomes, so homogametic XX, while the male has only 1 set. He's XO. In the sperm produced by the males, there's a 50-50 chance that they will have sex chromosomes in it, which will create a female, and there's 50% chance that it doesn't, and then it becomes another male. And we see this in grasshoppers, aphids, honeybees, and most of the bugs in the Hemiptera genera. Okay. Now, I think it's worth spending a little bit more time on honeybees because they're so unique in terms of their characteristics. Now, honeybee sex is determined by the number of sex chromosomes, but also by the environment in the hive as well. So we have our males, and these were the result of your unfertilized eggs. So they have one sex chromosome, what we know as monoploid, and then if they get fertilized, they become a female. Okay. So unfertilized eggs become male, fertilized eggs become females. So the females then have two sex chromosomes, but whether or not they become a queen depends on their diet. Queens are given a diet of royal jelly, which makes them sexually active, and sterile workers, remember bees, the females do vast majority of the work. Okay, the male's job is to just kind of tend the eggs and to go ahead and try and fertilize them. But the females go out and collect all of the honey, all of the nectar, and so all of the ones you see outside the hive are primarily female. So we start out with our queen, who's the only female that lays eggs. She's going to go ahead and lay eggs all over the place, and then the drones come along. If the drones fertilize them, they can become a female. If they are given royal jelly, that female will become a queen. But if the egg is not fertilized, it becomes a drone. Now, this is interesting if you have ever heard about like swarming techniques. So, most swarms, there is a female who's the active queen and another female who has been fed royal jelly and is ready to become a queen. So what happens is they fly out of the hive and they go ahead and battle, in some cases to the death. Once the battle is over, the winner can go ahead and take the hive back, and the loser, if they survive, can limp off and start their own hive. Now you may be thinking, how do you start your own hive if you're just one bee? Well, the queen can go ahead and lay eggs, and because there are no males to fertilize them, all of the new offspring will be male. And as long as we can do this fast enough, those males can now fertilize some of the eggs, producing females, which are now the workers that can go ahead and collect nectar and support the hive. So it's pretty fascinating when we start taking a look at honeybees. So the last thing I wanted to cover is the karyotype. Now, what is a karyotype? Essentially, it's a picture of the chromosomes arranged according to a standard classification with their numbers chromosome 1 through 22, and then down here at the bottom, your X and Y, which is your sex chromosomes. So autosomes for the vast majority of them, and then your little sex chromosomes down here. Remember, you should have two sets because you have a mother and a father. And we typically go ahead and do this right after DNA replication occurs and we start going into mitosis so that we can go have these nice little X's of the homologous pairs. Okay. So chromosomes are digitally arranged, so they're matched up with their homolog or partner chromosome. Homologous chromosomes are the same size and shape and carry the same genes, and one is inherited from each parent. While the chromosomes have the same genes, that does not, however, mean they have the same alleles. Okay, because remember, we can't see the alleles from a karyotype. They are numbered according to their size. We start with the biggest starting at 1 and end up with the smallest as we go down. And then we have the special sex chromosomes at the end. So let's take a look at some karyotypes. This is what we would see for a normal male. Okay, notice down here at the bottom right, we have this long chromosome and then the tiny little one. The long one here is the X. The tiny little one here is the Y. Remember, the Y is missing a whole bunch of information. Okay, and the number here is 2n, two sets of 23, which gives us 46 chromosomes. Now let's take a look at a normal female. It looks very similar to the male. The biggest difference here is in the sex chromosomes, where you have two X's and no Y. All right, but once again, you're dealing with a 2n number of 46. However, we also sometimes will see abnormal karyotypes. This is a good example as it has lots of abnormalities. Okay, these are what we would consider atypical. Okay, you can see that with chromosome 3, for example, there's a missing section on the bottom of the first chromosome. There's a missing section on the top of the third chromosome. Um, number 5 is missing a whole chromosome. It's mono, right, for that particular chromosome. Same thing down here with 22. And then we go ahead and take a look down here at the bottom right, and there's this ginormous chromosome, which is probably a fusion of some of the different chromosomes that are missing here. Okay, so those are the kind of things that you look for. So when we go ahead and take a look for an atypical, here's a new example. Can you identify how this is different from the typical, either male or female? So pause the video here, see if you can identify the atypical nature and whether or not you can tell if it's a male or a female. Okay, let's start out with gender. Can you identify the gender? Good, so this is a female because there are two X chromosomes and no Y. Now, what did you notice in terms of the chromosome structure? Yeah, hopefully you notice that there are little arrows pointing out to things in chromosome 1 and chromosome 12. So there seem to be some small abnormalities here, but nothing major. There are also two other major differences here. Did you pick those up? Yeah, 15 and 3 are completely missing chromosomes. And then number 13 doesn't have small differences, but there's a half of the chromosome that's just gone. So there are some pretty major differences from the traditional karyotype here. Let's give you another one and see if you can figure this one out. All right, so is this a male or female? Good. So this one's male because it has both an X and a Y. Did you figure out what was atypical? Take another second and find out what's atypical. Okay, what'd you find? Hopefully you identified that chromosome 4 and chromosome 11 has one of the homologs missing some sections. So these are the types of things that we can find when we go ahead and take a look at a karyotype. It's just one of many tools we can use to go ahead and do genetic counseling and to look for atypical situations within the genetic makeup of an organism. All right, guys, I hope you have a great rest of your day. Talk to you soon. Stay safe, stay healthy. Bye-bye.